Hi, my name is Chris Hardy, and I'm interested in studying obesity because it's one of the largest healthcare issues that we face today, and it's a growing problem. In just the last 30 years, the rates of obesity around the world have nearly doubled. And that's a huge problem because obesity is closely associated with things like diabetes and heart disease, which ultimately lead to a shorter lifespan. And the tendency of westernized societies to eat more and exercise less has always been a huge obstacle to treatment. But studies have shown that obesity is more than just what you eat and how often you go to the gym. There's a clear genetic component. And understanding what genes make you fat and how that fat affects the heart is the main focus of my research. So in order to address these questions, I use a very important model organism, the fruit fly, Drosophila melanogaster. And when I tell people that I study fat fruit flies, they generally think I'm crazy. But flies are much more similar to humans than people might expect, with about 75% of genes that cause the disease in humans also found in the fruit fly. You can even take fruit fly insulin and activate the mammalian insulin receptor and vice versa. So here at UNLV, we've established a very unique system in order to study obesity in fruit flies. It started about 15 years ago when my advisor, Dr. Alan Gibbs, took a set of fruit flies from the wild and brought them into the lab for a long-term evolution experiment. Each generation he would select for only the flies that could survive the longest without food, and these tended to be the ones that could store the most fat. So each generation, fat genes were passed down from generation to generation. It's gotten to the point now where 95 generations later, we've evolved fruit flies that are extremely obese. So fat that they even have a hard time trying to fly. And it's all purely genetic. They eat the same amount as their skinny controls. And so my first research question was to understand if they also suffer from heart disease. And they do. We took advantage of a machine that optometrists use to look at nerve structures in the eye, and we turned it around to look at heart function in the fly. You can see the results here. In a skinny, normal fly, the heart is open, fully contracts, opens, and fully contracts rhythmically over time. In our evolved fat lines, the heart is wider, it's dilated, and it's less contractile, making it less efficient. This is a condition known as dilated cardiomyopathy, similar to what you would experience in humans. We found that this is mostly caused by a lot of excess fat tissue that physically pushes the heart where it's supposed to lie in the animal down into the center of the animal. When we zoomed in on the fat tissue into the individual cells, we found that they contained extremely large lipid droplets, which are little compartments in the cell that hold most of the fat. The genes that control how lipid droplets are formed and how much fat they can store are very well conserved between flies and humans. And so the next step was to figure out what genes we'd selected for and what were causing these flies to become so obese. To do this, we formed a genome-wide association study where we took DNA from a skinny fly, DNA from a fat fly, and we sequenced it. So now we know all of the A's, T's, G's, and C's that make up a skinny versus fat fly. Using complex computer programs, we were able to find just over 1 million genetic differences, which you can see here, with the top 100,000 colored in red. We used these locations to identify a set of nearly 1,500 genes which were associated with muscle disorders, heart disease, and type 2 diabetes in humans. So we now have a set of genes that we can go through and test, and hopefully some of them will be targets for therapeutic intervention in the future. Thank you very much.